Hey everyone. Our group was the overactive adrenals. Um, and what we did when we first got assigned our unknown hormone, we compared the control data versus our unknown hormone data. And we looked at the measurements, identified any significant differences, and then we identified if it increased that measurement or decreased it by a significant difference. And here you can see there were significant differences in the body weight, the thymus, the tibia width, the ovaries, the uterus, the seminal vesicle, and the ventral prostate. And then the plasma levels increased at an of interest level. So not as much as a significant difference, but enough to catch our eye and make us think about it. So next we looked at the list of possible hormones that we were assigned, and we compared these hormones to the table of significant differences and went through each one and identified whether it could be that hormone based on a significant difference, or whether we could eliminate it based on significant differences or no changes at all. And so we'll go through those right now. So the first one we eliminated was the placebo. We just felt that there were too many significant differences for it just to be a placebo. And as you can see, body weight, thymus, tibia, ovaries, there were just too many differences, so we were able to eliminate that one right away. So next we eliminated thyroid stimulating hormone. We felt that the thyroid stimulating hormone would stimulate the thyroid gland and increase it in size if this was the hormone, and there was no difference in sizes between the control and unknown hormone. And also we believed that the thyroid counts per minute would increase because TSH would increase T3 and T4 levels, which would require more iodine, and there was no change there either. We also eliminated thyroxine. We felt that the thyroid gland would decrease in size if it was thyroxine because it would provide negative feedback to the hypothalamus and interior pituitary. And we would also expect iodine levels to increase, which they did not. Next, we eliminated glucocorticoids. We would expect blood glucose levels to increase if it was glucocorticoids, and there was no significant difference. And we would also expect the adrenal gland to decrease in size due to negative feedback, but there was no change in size of the adrenal gland. Next, we eliminated ACTH because we would expect the adrenal gland to increase in size, but there was no change in the adrenal gland size, and we would also expect an increase in blood glucose levels, but there wasn't an increase in blood glucose levels either. Next, we were able to eliminate the growth hormone, and this is because the growth hormone stimulates the thymus, and there was actually a significant decrease in size, and we would have expected an increase in size, so we were able to eliminate this. So next we eliminated somatostatin, and this is because we would expect the body weight and various other glands to decrease in size because this hormone inhibits growth hormone, but everything pretty much across the board increased in size. And we would also expect blood glucose levels to increase or decrease because this hormone can inhibit glucagon and insulin, but there was no change in blood glucose levels. So finally, we eliminated epinephrine. If it was epinephrine, we would expect higher blood glucose levels, but they did not change, and we would also expect the adrenal gland to decrease in size due to negative feedback, but the adrenal gland did not decrease in size. So this left us with 3 possible hormones that we wanted to get more information on to either confirm or eliminate. And those 3 are human chorionic gonadotropin, estradiol, and testosterone.
The first test our group decided on requesting were the vital signs. We chose to order the vital signs first because we wanted to compare the rats' vital signs to our expectations of how they would respond to excess amounts of testosterone, HCG, or estradiol. One thing we observed was that all of the vital signs increased compared to the control group. The increases in blood pressure, heart rate, and respirations all align with excess amounts of HCG or testosterone, but excess amounts of estradiol usually lead to a decrease in heart rate. We chose not to eliminate any of the hormones after this test because we wanted to confirm our thoughts on eliminating estradiol after receiving our second test. The second test we requested were the appearances and behaviors. We ordered this test because we knew it would help us decipher on whether we could eliminate estradiol. The dilated and reactive pupils and the increases in muscle size and tone both correlate with increased testosterone levels. Also, the reduced sleep, increased movements, and aggression also were expected. From this test, we were able to eliminate estradiol because these appearances and behaviors do not align with the increased levels of estradiol. The third test we requested was the radioimmunoassay of testosterone in the plasma. We chose this test because our data was all correlating and illustrating that the unknown hormone was likely testosterone, and we wanted to prove it. After analyzing our results, we found that there was a small amount of radioactivity counted in the experimental group. This meant that there was a larger amount of testosterone in the mixture from the experimental rats compared to the control rats.
As we requested our tests, we assessed what data would be of importance in deciding which hormone we think it is. After requesting the second test, appearance and behavior, we felt that we had enough data to request a radioimmunoassay. We looked at the increased mass for all 4 of the reproductive body parts, increased tibia length, and weight gain. We also looked at the decrease in the mass of the thymus, which we found may occur in the presence of testosterone, as well as glucocorticoids, but we had already ruled those out. The vitals indicated increased blood pressure, heart rate, and respiration rates. The appearance and behavior tests indicated increased muscularity and aggression. With all of this data, the radioimmunoassay was requested for testosterone, which indicated larger amounts of testosterone in the body than the control group. This, combined with the rest of the data, tells us that our unknown hormone is likely testosterone. This is supported by all of the data that was collected. However, there typically is a decrease in weight associated with testosterone. We feel that the reason we saw a weight gain, though, was that since the treatments were given to adolescent subjects, that the weight gain was due to increased muscle mass, giving them a more muscular appearance. The fact that the testosterone is a primary sex hormone explains why the reproductive parts increased in mass, and since it is an anabolic steroid, the muscle mass increased.